apologize to the people who've heard me many times, but I thought on this occasion I should perhaps present a little bit of a historical review of this work, since, as Giselle mentioned, it was initiated at the University of the Philippines. So I would like to talk about some very old work that was done before much of the audience was born. So what I'm going to talk about are our favorite group of organisms, the cone snails. And the cone snails are, of course, venomous creatures that are predatory, and different cone snails eat different prey. So this species will only eat fish, this species will only eat worms, and this species will only eat other snails. And what I'm going to show you is that cone snails have very complex venoms, typically with 200 different components, and that's what makes them so very interesting. And so I'd like to tell you a little bit about that today. However, let me review how this work started. It started in the 1970s when basically we wanted to know why this snail killed people. And so in the Philippines, there are a few stinging cases. And in the absence of medical treatment, 70% of stinging cases are fatal. And this is a very high number, considering that this snail is only about 5 or so inches long. So basically at the time, we had fairly modest aims. We simply wanted to know why this snail killed people. And at that time, at the University of the Philippines, in the College of Medicine, as it turns out, Department of Biochemistry, there's some members from that department here today, we didn't have very much equipment. And so I'd like to show you that even in the absence of equipment, it's possible to get started with science. I think many of the guys in this audience will at some point find yourself in a situation where you don't have a lot of equipment, but that doesn't mean that you should sit and do nothing. And this next slide shows you the first equipment we had in our lab. And it was the equipment that we used to assay for what were the components of the venom that probably killed people. Okay? And as you can see, it's not very complicated equipment, pretty standard to a laboratory. But the key player in this assay is the mouse. But of course, if you're in a medical school, the students are always doing nutrition experiments, and they always have controls. And so they're always generating mice that we could use for our experiments. And so we would put a mouse upside down on a screen. And in fact, when the mouse is on a screen, it can stay as long as it wishes. It's actually very good at hanging on to a screen. However, if we inject a component of the venom that's paralytic, then after a while the mouse would not be able to hang on to the screen anymore, because now its little toes would become paralyzed. And so as a result, the mouse would fall. And we call that the falling time. Okay? And that was the assay. That was the basis of all of our work on cone snail venoms in the first few years. So we simply looked for components of the venom that would cause mice to fall. And being chemists, we wanted to be a little more quantitative than to say that the mice fell. And so we actually measured what the falling time was. Okay? So the parameter that was measured in these experiments was to measure the falling time. And being chemists, we wanted to see the relationship between the falling time and the dose. So if you inject more, what happens to the falling time? Now, many of the people here in this audience have had some biochemistry, and I'm sure you've heard of Michaelis-Menten kinetics, if you've taken that in your biochemistry class. And it turns out that the falling time follows Michaelis-Menten kinetics. Now, those of you who had this biochemistry will recall that in enzyme kinetics you have what's called a double reciprocal plot, which is a straight line. And it turns out that if you plot the reciprocal of the falling time, which is the reciprocal of how fast things happen, versus 1 over the dose, you do get a Michaelis-Menten relationship. And so, although this is a very simple assay, it is in fact a surprisingly quantitative assay. And on the basis of the simple assay, we could quantitate how much paralytic activity there was in the venom. So why were we looking at paralytic activity? The reason we were looking at paralytic activity is because if you read the medical reports, the people who got stung by this snail, the reason they died was that after an hour and a half, they couldn't breathe anymore, and their diaphragms were paralyzed. And so what we reasoned was that the lethal components of the venom caused paralysis in humans, and therefore that those were likely to be the components that killed people. So we purified those components, and of course, by we, I mean Lily Cruz and myself. And once the components were purified, the components were sequenced by Bill Gray, who was a peptide chemist, and then chemically synthesized by Jean Riviere, who was a synthetic peptide chemist. And here were the two components in the venom that caused paralysis. And what you can see is that they are relatively small peptides, only 13 amino acids in the case of this, and 22 amino acids in the case of that peptide. And because we were able to synthesize these peptides, we could study what they did. And what we determined was that this small peptide, only 13 amino acids, really behaves very much like the deadly toxin you find in snakes, like alpha-bungeratoxin or cobra toxin. And so as you well know, if you get bitten by king cobra, you get paralyzed and you die, you can't breathe. And it turns out that this little peptide acts very much like the deadly toxin you find in cobra venom. And so as a result, it prevents communication between nerve and muscle. And this peptide by itself will cause paralysis. So we were surprised that this venom had a second paralytic component, because in cobra-like snakes, there's usually one major component. But in this venom, we found a second component, which was 22 amino acids, a little bigger. And when we figured out what this did, it turns out that this was similar to the toxin that you find in butete. Okay? So as you know, pufferfish are poisonous. You're not supposed to eat them. And the reason is that certain tissues in the pufferfish have a deadly toxin called tetrodotoxin. And what tetrodotoxin does is it wipes out all of the electrical signals in your nervous system. And we now know that that's because it inhibits voltage-gated sodium channels, which are responsible for the electrical signals in the nervous system. So tetrodotoxin is an alkaloid-like molecule, very organic, and so it was a surprise to find out that this peptide acted just like that organic molecule, despite the fact that it was so very different. And so the first thing that we discovered was that the active components that were present in the venom of Conus geographus were, in fact, small peptides. And ever since then, this has been the theme of the discoveries that have occurred in all marine snail venoms. So the reason why Conus geographus is able to kill you with such high efficiency is because when you get stung by the snail, it's a combination of being bitten by a cobra and eating a lethal dose of butete at the same time, right? And obviously that's not good for you, and that's why you get very high fatality rate. So we solved the scientific problem that we wanted to address. And so the question is, if we were successful in that, why are we still working on cone snails today? And the reason is that an undergraduate at the University of Utah, while we were working on this problem, made a very unexpected discovery. So if you take the venom of Conus geographus and you separate the venom out into size fractions, here's a fraction that has the peptides that are between 10 and 15 amino acids, and fraction B has the peptides that are about 20 amino acids. And now you take fraction B and you run it on a more sophisticated column. So by this time we had some equipment and we could separate things a little better. What you see is that fraction B is actually very complex. Each peak here is a different peptide. However, if we did the falling time assay, where we would inject the mice intraperitoneally, IP, then we only found 3 components that caused paralysis, and these were all variants of that sodium channel toxin that acts very much like the pufferfish toxin. And you can see there are lots of other peaks here, but because they were not biologically active, we didn't do anything with them because we didn't think they would be interesting. Then this young kid, he was about 18 years old, came to our lab and he said, you're doing it all wrong. You're injecting intraperitoneally, that's not how you should do it. You should inject each peptide directly into the nervous system, directly into the brain. And I wasn't so convinced that this would be a useful thing to do, and I tried to discourage him from it because I didn't think he'd learn anything. But the reason why the most creative research is done at universities is because the students do what they want, not what their professor tells them. And that really is a very important component of universities. The creativity of the students is what makes universities tick. So Craig, despite my objections, took each peak here, injected it into the brain of a mouse, and he got the following spectacular results. This is the same chromatogram. Here are the paralytic peptides, but now here's a peptide that made mice jump and twist as they were jumping. This made mice uncoordinated. This big peptide put mice to sleep. So the mice would be asleep for 12 to 24 hours and then they'd wake up and they were perfectly fine. But that was only true if the mice were under 3 weeks of age. If they were over 3 weeks of age, instead of going to sleep, they'd run from corner to corner, climb the sides of their cages. So suddenly one went from sleep to hyperactivity. This peptide made them drag their back legs. This peptide made the mice run around in circles for hours. This made them swing their heads back and forth. This made them kick on their back and scratch. This made them depressed. This made them comatose. This caused paralysis. This caused trembling. This caused scratching. This caused convulsions. And a few peptides didn't seem to do anything. But as you can see, the astonishing result is that every peptide that we tested from this one venom caused a different behavior when they were injected into the central system, nervous system of the mouse. So this simple experiment told us that the venom of a cone snail was not a mixture of a few paralytic toxins, but was this extremely complex pharmacological mixture. And so ever since then, we've been trying to understand why this complexity. So what I'd like to do today is to bring you up to date on our understanding of why these venoms are so complex. If you take a sea snake venom, 90% of a sea snake venom is a single component, which is a paralytic toxin. In contrast, here you can see this is only one of 4 fractions from a single venom. This is the peptides that are about 20 amino acids in length. And so in fact, this venom has about 200 different components. So first of all, what do these snails do with their venom? So this is a fish hunting cone snail, and this is a real-time video of what the snails do. And so you're going to see this is a fish hunting cone snail. So there's its prey, it sticks out its proboscis, and you're about to see it attack the fish. And as you can see, this all happens very fast. Notice the fish has very stiff fins. And so that's how these snails get their meal. Okay, so that's a fish hunting cone snail. Okay, so the snail has an organ that has about 50 to 100 teeth, and when it goes hunting, it moves one of these teeth to its proboscis, and as you saw, it can extend the proboscis very far. This duct is where the venom is made, and now this muscular bulb is like the bulb of a medicine dropper, and so it presses out the venom through the proboscis, through the tooth, and the tooth, it turns out, here's the tip of the proboscis, and so the venom flows here into the prey, and you can see at the end of the tooth is a hole, so this is actually not just a harpoon-like tooth, but also a hypodermic needle, and you can see it's barbed, so it's shaped like a harpoon, so this is both harpoon and hypodermic needle. So about 55 million years ago, this group of snails evolved what's essentially a drug delivery device. It's the equivalent of disposable hypodermic needles, right? They only use this once, and then they throw it away. They keep on making them. And because they now have a way of injecting their venom into the prey, this is no doubt partly responsible for their success. And if we look at different species of cone snails, we find different designs for the harpoon. So you can see this is a very sophisticated delivery device. So the first thing we did to try to understand why this venom was complex was to simply ask the question, okay, if you have a snail that eats fish, what components of the venom will paralyze a fish? And typically you get 5 or 10 different components of the venom. Here's examples from a single venom that all paralyze a fish. And if you ask where they all act at what's called the neuromuscular junction. So when you move your little finger, your brain sends electrical signals to the nerve. The electrical signals come down the nerve, and as a result of that electrical signal, calcium channels open up, let calcium into the end of the nerve, and as a result, you get exocytosis, the fusion of these vesicles loaded with chemicals with the membrane, and so those chemicals, the neurotransmitter, is released into the space between nerve and muscle. And it turns out the snails have one venom component or more that blocks these calcium channels. And so this is paralytic because now the nerve cannot signal the muscle. Now, if the nerve releases the neurotransmitter, that binds to the receptor on the muscle, the postsynaptic side, and it's called the acetylcholine receptor, and that receptor causes an ion channel to open that lets sodium across the membrane. That changes the voltage across the membrane and starts an action potential which is carried by the sodium channels. And the snails have peptides that inhibit this postsynaptic receptor so that even though there's acetylcholine, these remain closed. And just for good measure, in addition to wiping this out and wiping this receptor out, they also wipe out this sodium channel that carries the electrical signal. So any one of these is sufficient to paralyze a fish. So why are they making so many? So many millions of years ago, cone snails also discovered a principle that medical practice has only recently discovered. It's better to combine multiple pharmacological agents to have a physiological effect than use a single one. So today, if you want to cure AIDS or you want to cure cancer, you don't just use one pharmacological agent, you use a combination. So the principle of combination drug therapy was essentially discovered by these snails many millions of years ago. And so whenever the snails do anything to another organism, we have found that they always use multiple pharmacological components, not just one. So what we call groups of toxins that act together for the same physiological endpoint is cabals. Because cabals are secret societies that are out to overthrow existing authority. And so toxin cabals are out to overthrow the normal physiology of the snake. So we call this group of peptides that essentially prevent communication between the motor nerve and the muscle, the motor cabal. And so we were pretty proud of ourselves that we figured out all of these peptides and what their targets were. But in fact, there's a problem. You can take any one of these toxins, or all of them in combination, and inject it into a fish, and it takes you 20 or 30 seconds to immobilize the fish. And yet, if you looked at that video, it took approximately 1 second. Furthermore, if you take these peptides that we isolated from the venom and inject it into a fish, the fish looks like this. All its musculature is relaxed. However, if you look carefully at the fish when it was injected by the snail, the fish looked like this, with all of its fins very, very stiff. So what's going on here? What's going on here is that in addition to the motor cabal, there is a second cabal, which we call the lightning strike cabal. And it's as if the fish had been tasered or electrocuted. And so what I'd like to show you are the main components of the lightning strike cabal from a particular venom. And the two main components we call kappa toxins and delta toxins. And what I'm going to show you is how these act. So here's a fish, and here's the nervous system of a fish with action potentials going through the nervous system. If we look at one axon, the excitation goes through an axon transiently, and the reason is that sodium channels open up as an action potential approaches and the membrane changes, and the sodium channel, when it opens, it specifically only lets sodium ions across the membrane. So this opens up, sodium ions enter, and then later potassium channels open and they let potassium out. Okay? Now that combination guarantees that the excitation is transient. So how does the snail paralyze the fish? And it does so because it has a specific toxin that hits the sodium channel. Normally a sodium channel will open, and very quickly afterwards it will close again. But when this toxin binds, it remains open. So sodium keeps on flowing into the sodium channel. Now normally the potassium channel helps to end the excitation, but the second toxin blocks the potassium channel. So what happens? That means that this axon remains excited all the time. It's almost as if you electrocuted the fish at the point where the venom was injected. And so that's why you get this very, very stiff fish. So why does this happen so fast? The reason this happens so fast is in contrast to the other toxins which have to go through the circulation, spread through the body of the prey to paralyze the muscles. These toxins act right at the site of injection. All they have to do is bind to the closest nerve, and you get this very effective immobilization. So what I'd like to show you are the components of the lightning strike cabal, and we've worked out a few more, and the components of the motor cabal. So if the lightning strike cabal is so effective, why do you need two cabals? Okay? So this is now a biological question. Why did the snail evolve two different cabals when it seems that one cabal should be enough? And what I'd like to do to answer that question is show you another video. So look carefully here. This is Conus bulliatus, and there's the proboscis. It's about to inject the fish. Look how fast that was. And look at how stiff the fins are. You can see this. So that's the lightning strike cabal in action. From the injection site, all the nerves are firing, and that's why it's like a tetanic paralysis. Okay? And so you can see this is very effective. So why do you need the other cabal? Well, this shows you why. Remarkably, this fish is going to recover. Okay? And in this particular case, of course, it's too late. The fish is not going to escape. However, in an aquarium, when these snails harpoon a fish, then if the fish is big enough, sometimes when it recovers that way, the fish can tear itself loose from the harpoon. And under those conditions, what we find is that the snail will always do a little search pattern looking for a paralyzed fish. So in the real world, it pays off to have 2 cabals. Okay? In the lab, it looks like, okay, 1 cabal should be enough. But in the real world, the chances of success are much increased by having 2 cabals. So in the real world, to be effective, if you're using a chemical strategy, these strategies are much more sophisticated, I think, than any of us would have predicted. Okay? And evolution results in a very high level of kind of sophistication that we would never have predicted. And so the venom, the other thing we found is that when you go from one conus species to the next, you find that there's no overlap. At the molecular level, each species has its own set of peptides. And that's a lot of peptides, 200 different peptides. So what's going on here? We understand at the molecular level how this happens. So a peptide is generated from a larger precursor, a pre-propeptide, and this shows you an example of one such sequence. And there are gene families that encode peptides that have different shapes. Within one gene family, what you find is that there are conserved cysteine residues that can form cross-links, disulfide bonds, and these are conserved within one gene family. So in fact, what's going on here is within one gene family, the shape of all the peptides is approximately the same. However, this is like the antibody system in which there are conserved and hypervariable regions, and so the cysteine residues are conserved, but all the amino acids in between are hypervariable, and as a result, these peptides change their sequence very, very rapidly as these snails evolve. So, first of all, why are there so many different peptides in one venom? And why do the venoms of different cones have different peptides? Well, we found that the venom is not just used to capture prey, but also to defend against various predators and to deter competitors. Okay? So in other words, all of the biological interactions of these snails with their predators, with their prey, and with their competitors are really through these venom peptides. And so there are lots of organisms that they have to interact with. So then the question is, so that's why there's so many peptides in the venom. So you've seen to capture prey, maybe it takes 15 peptides, but why there are 200? Presumably the rest are to defend themselves against predators of their own and also for competitive interaction. Now the question is, why do you have to change the peptides when you go from one species to the next? Why do these evolve so quickly? And our answer is an ecological one. That in fact, if you look at a species, every species has its own ecological niche. And what that means is that each species has its own spectrum of prey, its own predators, its own competitors. And so every time you have a new species, that has different prey, different predators, and different competitors. And so as species are generated, what you get is an evolution of fitness, and these are the genes that have to change most quickly when you go from one species to the next. And so as a result, each species has multiple biotic interactions, but because it's occupying its own particular ecological niche, that gives selective pressure to these genes to evolve very, very quickly. So we're pretty excited that pretty soon we'll have probably 2 genomes of conus species to look at, and so we're going to see how these particular genes evolve, and so we're really looking forward to understanding that. And we call these genes that don't act within the organism that makes them, where the gene product acts somewhere else, exogenes, which we expect to be very rapidly evolving. Now, what does this evolution really mean in practical terms? So if you look at fish hunting cone snails, this is one group of fish hunting cone snails, and all of the ones that I've shown you are in this branch. But in fact, there are 4 groups of fish hunting cone snails. And if you look at this group here, that's quite closely related, what you find is, although these species eat fish, they have a very different strategy. So this shows you Conus geographus itself. If you drop a fish in, instead of sticking its proboscis out, these snails open their mouths, and you can see they have a very big mouth. And in fact, you might say they have a gargantuan mouth. And so that's their behavior. If we look at another species in this branch, Conus tulipa, this has hairs at the end of its mouth, and you can see it always engulfs the fish first before it stings it. So what's going on here? So these types of snails live close to coral reefs, and at night they come out and they look for small schools of fish that are hiding in the coral reef, swimming in the coral reef. And long ago, these snails discovered there's more than one way to catch a fish. You can use a hook and line strategy, like the first group of cone snails I showed you, or you can use a net strategy. And you can, if you're clever at it, you can bag a whole school and then begin to pick them off one by one. Now, if you're using this strategy and you had a lightning strike cabal, then what would happen is, when you harpoon the fish, it would be twirling around at the end of your proboscis with very stiff fins. And if that fish is already in your mouth, that's not a good idea, because you might get wounded. And so in this type of snail, we do not find the lightning strike cabal. Okay? That's absent. Instead, the snail releases a little bit of its venom, and as a result, the fish become lethargic. They act as if they're spaced out. And so we call that, instead of the lightning strike cabal, we call that the nirvana cabal. And so the nirvana cabal makes this fish very, very lethargic and spaced out. And they also have a motor cabal. So what we've done is we've analyzed the components of the nirvana cabal, and we've isolated a bunch of peptides. And what all these peptides do is they quiet down neuronal circuitry. And so we were wondering if they're quiet down neuronal circuitry, can we use them in pathological conditions where you have overactive neuronal circuits? So what are 2 pathological conditions that you can think of where you have overactive neuronal circuits? One of them obviously is epilepsy, where you have the nervous system too active, and that's why you get a seizure. And surprisingly, another is chronic pain. So your pain circuitry gets overactive and you feel pain all the time. Okay? Well, it turns out that 2 components of the nirvana cabal have promise. One of them for curing pain, and one of them for curing epilepsy. And so both of these peptides that came from cone snail venoms, that shows you their structures, have reached phase 1 clinical trials, and hopefully they'll be developed further. There is, however, so here are a whole bunch of conus peptides that have reached clinical trials for pain. 4 of these were discovered in our lab, and 2 were not. And you can see one of them has been approved, called Pre-Alt. And let me tell you how Pre-Alt was discovered. It was discovered again by an undergraduate who looked at this snail, Conus magus, and he found a peptide that caused shaking, which he called the shaker peptide. So that's how we discovered the peptide. And sure enough, Michael was able to sequence the peptide, and this is the sequence of that peptide. And today, it's a drug for intractable pain. Okay? So it's a commercial drug. It's been approved by the FDA in December 2004. Now, if I can take a few more minutes, what I'd like to show you is, first of all, why this snail has evolved this peptide, and secondly, why it's a drug for intractable pain. So let's go back to our fish. Conus magus is a fish-hunting cone snail. And so here again you see the nervous system of the fish. And now let's look at the junction between nerve and muscle. So this is striated muscle, and this is the motor nerve. And if we zero in on the synapse, the neuromuscular synapse between nerve and muscle, essentially an action potential coming down the nerve is always transmitted across the synapse to the muscle. So a motor action potential in the nerve results in calcium channels opening up. And so as a result, calcium enters the nerve. The entry of calcium, elevated calcium, then causes these vesicles to fuse, releasing their chemicals, the neurotransmitter acetylcholine. And in the normal course of events, acetylcholine binds the acetylcholine receptor, and that opens up the ion channel. And as a result, that depolarizes the muscle, and so you get an action potential in the muscle. So an action potential in the nerve normally always results in an action potential in the muscle. So why did the snail evolve this peptide? Well, this peptide is part of the motor cabal. So this peptide, Pre-Alt, very specifically binds to those calcium channels. And so what happens, the action potential reaches the end of the nerve, but because the calcium channels are plugged, you don't get calcium going into the nerve, you don't get the release of neurotransmitter, and therefore this causes paralysis of the fish. Okay? So that's the reason the snail has evolved this peptide. It's a very specific peptide that binds these calcium channels at the ends of nerves. So normally, an action potential in the nerve always results in an action potential in the muscle, but not in the case where you have Pre-Alt. Well, what happens in the mouse? What happens in the mouse is that the same thing occurs. An action potential on the nerve always is transmitted, but there's a difference here. And that is, these calcium channels are not green, they're red, and they're Pre-Alt resistant. And so even though if Pre-Alt is present, calcium channels are not blocked, and so the normal things that happen when an action potential reaches a nerve happens, and so now you get an action potential in the muscle. So for mammals, this peptide is not paralytic. And so when you swallow this peptide, you don't get paralyzed because you have the red calcium channel. However, mice and we also have the green calcium channel, but it's in a different place. It's in the pain circuitry. And so when you're feeling pain, your pain fiber keeps on firing, and in order for that signal to be seen by the central nervous system, there's a synapse between the pain fiber and a cell in the spinal cord. And now, because the calcium channels there are green, they're plugged by Pre-Alt. And so even though you have very severe pain and your pain fibers are therefore firing constantly, because you have the synapse completely inhibited, as a result, you do not perceive the pain. So you can see that this will work as an analgesic. Now, I think I'm being told that my time is up, but what I'd like to emphasize here is an important scientific point, which is that if you look at all nervous systems, whether they're in fish, in frogs, in us, or even in worms and in Drosophila, these calcium channels look very, very similar. Okay? So the one in fish, the green calcium channel, the red calcium channel, you can tell them apart right away. However, what I've shown you is that although the types of calcium channels look very similar, and in fact the peptide always recognizes only the green one, not the red one. Okay? Whether it's in fish or in rats or in frogs. But the difference is that in people, this green calcium channel is only found in the pain circuitry. The green calcium channel in fish is found at the neuromuscular junction. So although structurally similar, they're not similar at all in their expression pattern. So I think that shows you the promise of the components of these venoms for future drug development. Now, I'm running out of time, and I don't want to go over, but one thing I did want to show you, this is the reason why this is a drug. If you take morphine, morphine's a great drug, and it's far cheaper, so why not take morphine? Well, the problem is that if you have intense pain, and this is a measure of pain, and you take morphine, you decrease the pain. You take the peptide, you decrease the pain. So why not just take morphine? Why bother with the peptide? Well, many of you may know that the trouble with morphine is if you have to keep on taking it, after a while it doesn't work well anymore. And this is a phenomenon called tolerance. And so what you can see is that after 7 days in this animal model, morphine doesn't work anymore, but the peptide works just as well. So for patients who are suffering continuous pain and who are now tolerant to morphine, this becomes one of the choices to alleviate their pain. Okay, what I'd like to end up with, and I'll do this very fast, is to tell you that cones are not the only venomous snails. The other venomous snails are turids and auger snails, and we have a collaboration with the lab of Giselle Concepcion and others to look at turids. Why are we interested in turids? Because in contrast to cones, where there's 500 to 700 species, there are 12,000 species of turids. So there are many, many more. Now, in the old days, you couldn't work on turids, okay? And the reason was they tend to be very small, and there's not a lot of them. But with modern biology, today, you can access a very large number of these species. And so we're beginning to do that. I'm not going to show you another video, but this shows you how small some turids are, a few millimeters in length. And we can catch them in these specialized nets called lumen-lumen. This shows you the relationship between the cone snails and the turids. And so these snails are a few millimeters long, but what we've discovered is that their venoms, even though they're small, are just as complex as cone snails. They have a few hundred peptides in their venoms. So this is a completely unexplored area. Most of these turids haven't even been named yet. Here, for example, are a bunch of turids that are about 5 millimeters long. But using molecular biology, today you can access what each of these different species of turids are making, and that indeed is what we're trying to do in a collaborative project with the University of the Philippines, with Giselle's lab, Frank's lab, and others at the university. And so here are some sequences that we get by cloning, and so what we're doing is we're synthesizing all of these peptides, and now we'd like to know what each of them do. So there's a lot more work to be done here. And we estimate that there may be as many as 2.5 bioactive compounds in the venoms of these turids. So I'd like to end up by just acknowledging this very large collaboration, and I'm very happy that several of my collaborators at the University of the Philippines are in this audience, Miran and G, Frank, and of course Giselle. In Utah, this is our collaborative group, and we collaborate with electrophysiologists in Germany, biodiversity people in Paris, and also in Switzerland and in Australia. So thank you very much for your attention, and I'd be happy to